So picture this. You're sitting in a really comfortable chair, right? And you're intensely focused on reading a novel.
Oh yeah, the classic hyperfocus state.
Exactly. You are completely oblivious to the world around you, the hum of the refrigerator, the cars driving by outside, the ticking of the clock. It is just all tuned out,
Right, you're locked in.
completely unbreakable hyperfocus. But then suddenly, a super loud crash comes from the kitchen.
Boom.
Right. And in a fraction of a millisecond, your concentration just entirely shatters.
Yep, it's gone.
Your eyes dart up, your heart rate spikes, and your brain just, well, entirely shifts its processing power to this new, potentially vital piece of information. I mean, it's an everyday scenario, but what is physically happening inside your skull in that exact millisecond?
It's a profoundly relatable experience, you know. But underneath that seemingly simple split-second shift is just a staggering symphony of neural computations. Yeah, I mean, we are talking about massive brain-wide networks evaluating your environment, calculating probabilities, and physically hijacking your body's sensory organs.
Wow.
And for decades, neuroscientists have been trying to map exactly how that invisible machinery actually works.
And that is exactly what we are doing today because, when we talk about something like a broken bone, we can just look at an X-ray.
Right, it's highly visible.
Yeah, it's structural, it's binary. You point to a jagged white line on the scan and say, there's the problem. But when we step into the world of human attention and cognitive control, that X-ray machine is entirely useless.
Completely. You can't photograph a thought.
Right. We are looking at a landscape of dynamic, invisible networks that are constantly shifting, communicating, and reconfiguring in real time. So, welcome to today's deep dive, tailored specifically for you, the learner. Today, we are on a mission to unpack the precise, cutting-edge neuroscience of how you pay attention and, honestly, more importantly, how you control your own behavior.
It's a massive topic.
It really is. We're going to explore the structure and function of two fundamental systems. The Dorsal Attention Network, which we'll call the DAN, and the Ventral Attention Network, the VAN.
The classic duo.
Yeah. And then we'll look at how these are governed by higher-order cognitive control networks, like the frontoparietal control network.
Right. And to do this properly, we are relying on a truly remarkable stack of source material today.
Oh, yeah, we have some heavy hitters.
We really do. We're drawing from highly influential, peer-reviewed neuroscience papers. And this ranges from the foundational texts that quite literally define the field of attention, all the way up to cutting-edge 2024 reviews from top-tier journals like Nature Reviews Neuroscience, PNAS, and Cerebral Cortex.
Which is just incredible access to the latest data.
Exactly. We aren't just looking at the broad strokes of what these networks are. We're diving deep into the structural changes, the intricate network connectivity, and the regional activations that explain how cognitive control and executive function actually operate on a biological level.
Okay, let's unpack this, because before we can understand how the brain, controls our thoughts, what we call cognitive control or executive function, we first need to understand the fundamental hardware of how the brain attends to the world in the first place.
The foundational layer, yeah.
Right, which brings us to the duality of attention, the DAN and the VAN. So let's start with the Dorsal Attention Network. When I'm sitting there, deeply immersed in that novel, reading line by line, what exactly is the DAN doing?
Well, so when you are deeply immersed in that book, your dorsal attention network is basically the primary engine running the show.
Okay.
The DAN is responsible for top-down, goal-directed spatial orienting.
Top-down spatial orienting.
Right. And to really trace this back to the foundational literature, we have to look at Posner and Peterson's seminal 1990 paper in the Annual Review of Neuroscience.
The 1990 paper.
Yeah, it was titled The Attention System of the Human Brain. And we also have to look at Fox and colleagues' 2006 paper in PNAS, Spontaneous Neuronal Activity Distinguishes Human Dorsal and Ventral Attention Systems.
Why were those papers so pivotal?
Well, because before this work, attention was often thought of as just this diffuse general property of the whole brain, like a fog that just kind of covered everything.
Oh, I see. Just a general state of being awake, basically.
Exactly. But these papers laid the groundwork for understanding that attention isn't just one monolithic thing. It is divided into highly specific, anatomically distinct systems.
So geographically speaking, where is this network? Like, how does it actually execute that top-down goal of reading the book?
Anatomically, the DAN is distributed across both hemispheres of your brain. It's primarily anchored in the intraparietal sulcus and the frontal eye fields.
The frontal eye fields. That sounds very literal.
It is. And what's crucial to understand about the DAN is that it's closely tied to sensoromotor regions. It utilizes what we call spatially organized receptive fields.
Okay, let's break that down for a listener. What does a spatially organized receptive field actually mean in practice?
Think of your visual field like everything you can see in front of you right now as a massive grid.
Okay, I'm picturing a grid.
The neurons in the DAN are physically mapped to correspond to specific coordinates on that exact grid.
Wait, really? Like an actual map?
Yes, exactly like a map. It's highly active during deliberate shifts of attention to salient objects in your external environment. And it even guides visually directed reaching actions, like grabbing your coffee mug.
Wow.
So when you were reading, the DAN is taking your internal goal, which is, I want to read the next sentence, and it's translating that into physical coordinates.
So it's telling my eyes where to go?
Precisely. It sends signals to your frontal eye fields to literally move the muscles in your eyes, tracking from left to right, line by line.
That is wild.
And at the exact same time, it's tuning your visual cortex to enhance the processing of those specific black letters on the white page, while simultaneously suppressing the visual noise of the table or the room around you.
Okay, so the DAN is essentially the ultimate top-down boss of spatial focus. It's like, well, it's like a heavy freight train on a track.
A freight train, I like that.
Yeah, because it has a schedule, it has a destination, and it is moving purposefully forward, laying down the tracks of your attention exactly where your goal dictates they should go.
That's a great way to visualize it.
But then the loud crash happens in the kitchen. Now, listening to a crashing plate is definitely not part of my goal to read the novel. So that is where the Ventral Attention Network, the VAN, enters the picture, correct?
That is exactly where the VAN steps in. If the DAN is your goal-directed focus, the VAN is concerned with the entirely non-spatial aspects of attention.
Non-spatial, okay.
Right? It's your brain's intrinsic alerting system. It governs bottom-up, stimulus-driven orienting.
So it reacts to the world rather than imposing a goal on the world.
Exactly. When an unexpected, highly salient event happens in the environment, like that loud crash, the VAN completely takes over.
How does it do that?
Well, a fantastic way to conceptualize the mechanics of this comes from Menon and Desposito's 2022 paper in Neuropsychopharmacology. It's titled The Role of PFC Networks in Cognitive Control and Executive Function. In that paper, they actually adapt some concepts from Corbetta and Schulman's highly influential 2002 Nature Reviews Neuroscience paper. And they outline the VAN's critical function as a circuit breaker.
A circuit breaker. Oh, I like that terminology. So if the DAN is the train on the track, the VAN is the emergency brake that gets pulled if a car stalls on the tracks up ahead.
Exactly.
It overrides the train's momentum to prevent a disaster.
That's exactly it. The VAN literally acts as a circuit breaker on the DAN. It abruptly halts your top-down, goal-directed reading process to forcibly direct your attention to the exogenous salient event.
And where does the VAN live in the brain?
Anatomically, unlike the DAN, which is bilateral, meaning it's on both sides, the VAN is strongly lateralized to the right hemisphere of the brain.
Just the right side?
Mostly, yes. It's anchored in the temporal-parietal junction and the ventral frontal cortex.
But wait, I have to stop you there because this is where I think a lot of people get tripped up.
Sure. What's the hang-up?
If the VAN is just automatically reacting to a loud noise in the environment, how is that any different from a simple, hardwired physical reflex?
Ah, I see what you mean.
Like, you know, when a doctor taps your knee with a rubber hammer and your leg kicks, that doesn't involve attention in a higher order cognitive sense. It's just a spinal reflex. So is the VAN doing real computation or is it just a glorified biological tripwire?
That is such a good question, and honestly, that's exactly what a lot of early researchers assumed, but it is entirely incorrect.
Really? So it's not a tripwire?
Not at all. The VAN is not a blind, hardwired reflex. It is performing highly sophisticated, real-time cognitive evaluation.
How do we know that?
To understand why, we have to look at Vossel and colleagues' 2012 paper in the Journal of Neuroscience. It's titled Deconstructing the Architecture of Dorsal and Ventral Attention Systems with Dynamic Causal Modeling.
Okay, dynamic causal modeling. That sounds incredibly dense.
It is fairly dense math, yeah.
How do they actually prove the VAN is doing math and not just flinching?
Well, dynamic causal modeling, or DCM, is a mathematical framework used to infer the hidden architecture of neural networks.
Hidden architecture?
Yeah. Usually in brain imaging, if area A lights up and area B lights up at the same time, we just say they're correlated.
Right. They happen together.
Exactly.
Yeah.
But correlation doesn't tell us who is giving the orders.
Yeah.
Area A might be talking to area B, or area B might be talking to area A, or hidden area C might be shouting at both of them simultaneously.
Oh, that makes sense. Correlation isn't causation.
Exactly. So DCM uses differential equations to model how a change in the neural state of one region directly causes a change in another region. It actually maps the direction of the flow of information.
Oh, wow, that is fascinating. So it's not just looking at a map of traffic, it's looking at which car is actually pushing the other cars out of the way.
That's a perfect way to put it.
So what did Vossel's team find when they applied this traffic pushing math to the Dan and the van?
They proved that the van isn't just a passive alarm bell. It's a complex cortical network that is in constant dynamic communication with the Dan. The van doesn't just react blindly to any noise. It actually evaluates the salience of the noise based on context.
Wait, so it knows the difference between a crash that matters and a crash that doesn't?
Precisely.
Yeah.
Let's say you're sitting on a screeching subway car trying to read a book. The van might completely ignore a loud metallic clank because contextually, the brain has already predicted that noise.
Right, you're on a train. Clanks happen.
Exactly. It's not novel. It's not biologically relevant to your survival, but a sudden loud crash in your perfectly quiet kitchen. The van rapidly computes that this is highly anomalous and potentially dangerous.
So it does the math.
It does the math. It decides the noise is salient, breaks the Dan's ongoing circuit, and essentially hands over a set of crude spatial coordinates so your Dan can quickly take over again and physically orient your eyes toward the kitchen to investigate. Yeah, they are two separate frontoparietal cortical systems,
Right.
but they cooperate seamlessly to produce normal adaptive behavior.
Okay, so the emergency brake is smart. It knows when a shape on the tracks is just a shadow and when it's an actual stalled car.
Exactly.
But this raises a massive logistical problem in my mind.
What's that?
Well, if the Dan is the worker on the factory floor executing the focus, and the van is the safety officer on the floor breaking the focus when necessary, who is the manager? Like, who is sitting in the corner office telling the Dan what the ultimate goal is in the first place? Or telling the van what kind of environment we are currently in so it knows what to ignore?
You just hit on the exact transition point in neuroscience from studying pure attention to studying cognitive control and executive function.
Okay, lead the way.
The managers in this scenario are the cognitive control networks, specifically the frontoparietal control network, or the FPCN.
The FPCN.
Right. And for a long time, cognitive control was viewed as this single monolithic construct. We knew it was housed somewhere in the prefrontal cortex, the highly evolved front part of the brain right behind your forehead. But we treated it like one giant general-purpose switchboard.
Right, the classic idea of the executive part of the brain. Just one big boss making all the decisions.
Exactly. But the field underwent a massive conceptual shift. In 2008, Dosenbach and colleagues published a landmark paper in Trends in Cognitive Sciences called A Dual Networks Architecture of Top-Down Control.
Dual networks. So two managers.
Yes. They look at massive amounts of functional MRI data and realize that the prefrontal cortex is not one big switchboard. They introduce the idea that cognitive control is actually supported by a dual network architecture.
How are the two networks different?
They separated the rapid, adaptive, moment-to-moment control handled by the FPCN from the stable, long-term, set maintenance control handled by other networks, like the cinguloopercular network.
Okay, so we went from thinking there was one manager to realizing there's actually a hierarchy of management.
Right.
But the FPCN itself, the network responsible for that rapid adaptive control, it isn't even a single entity, is it? Because this is where it gets really interesting in the newer research.
Oh, you mean the subsystems?
Yeah, when we dig into the more recent literature, it turns out this specific manager has distinct personalities or sub-departments that are completely walled off from each other.
Yes, the FPCN is incredibly heterogeneous. And to really explore the depths of this, we have to deep dive into a brilliant 2018 paper by Matthew Dixon, Alejandra de la Vega, and their colleagues.
Right, the PNAS paper.
Yep, published in PNAS. It's titled Heterogeneity Within the Frontoparietal Control Network and Its Relationship to the Default and Dorsal Attention Networks. The researchers basically wanted to know if this control network was truly a generalist or if it had specialized divisions.
And how do you even test for that? I mean, if it all looks like one single network on a standard brain scan, how do you mathematically prove it's actually two?
They used incredibly sophisticated mathematical tools, specifically hierarchical clustering and machine learning classification analyses, to examine the functional connectivity patterns across multiple cognitive conditions.
Okay, let's ground that jargon for the listener. What is hierarchical clustering doing to a brain scan?
Think of it like analyzing a massive high school cafeteria.
Okay, I can picture that.
Ah,
Lots of noise.
right. If you just looked in the balcony, it looks like one giant chaotic mass of students. But if you track who talks to whom, who trades sandwiches, and who arrives together over the course of a month, you can start grouping them.
You find the clicks.
Exactly. First, you cluster them by the tables they sit at. Then, you look closer and cluster them by subcultures. The athletes, the theater kids, the debate team.
Oh, I see.
The algorithm does this mathematically with tiny 3D cubes of the brain called voxels. It tracks the electrical blood flow signals over time and says, these 1,000 voxels are firing together in a slightly different rhythm than these other 1,000 voxels.
So they let the machine learning algorithm watch the cafeteria of the prefrontal cortex. And what did it find?
What they found completely shattered the idea of a unitary domain general system. The algorithm proved that the FTCN is actually split into two distinct subsystems, which they simply named FPCNA and FPCNB.
A and B. Nice and simple.
Very straightforward. They are anatomically interleaved, meaning their regions are physically right next to each other like a mosaic across the lateral prefrontal cortex, the parietal cortex, and the temporal lobe, but functionally. They're communicating with entirely different parts of the brain.
And their distinct roles are absolutely fascinating. If we think of FPCNA and FTCNB as two different project managers sharing the exact same office, their desks might be right next to each other, but they are looking out of completely different windows.
That is a perfect analogy. The Dixon 2018 paper detailed how these two subsystems exhibit radically different patterns of global connectivity. FPCNB exhibits much stronger functional connectivity with the Dorsal Attention Network. Yes, RDAN. The top-down spatial focus train we talked about earlier. Because of this structural and functional alliance, FPCNB is preferentially involved in regulating visuospatial perceptual attention.
So it manages the outside stuff.
Exactly. It's the manager tasked with handling how you interact with the external physical world.
So FPCNB is the manager looking out the window. It coordinates with the DAN to help you read the book, drive a car, or catch a baseball. It's entirely outwardly focused. What about FTCNA?
Conversely, FPCNA connects strongly with what we call the default network, or DN.
The default network.
Now, the default network is famous in neuroscience for its role in introspective processes. It is the network that comes online when you are daydreaming, mentalizing about what other people are thinking, engaging in autobiographical memory, or just experiencing spontaneous, unguided cognition.
So inner thoughts.
Right. Therefore, because of its tight linkage to the default network, FTCNA regulates introspective, self-generated thought.
I love this because it explains so much of our daily human experience. FPCNA is the project manager looking inward. It is the one coordinating with the default network to help you imagine what you are going to say in a stressful meeting tomorrow, or reminiscing about a childhood vacation while you were just staring blankly at your computer screen.
Exactly. It is a highly specialized evolutionary division of labor for cognitive control. You cannot effectively manage complex external stimuli and complex internal rumination using the exact same neural pathways without causing a massive bottleneck.
It would just be a traffic jam.
Precisely. The brain solved this by duplicating the control network and specializing one for the outside world and one for the inside world. And this twofold differentiation was remarkably robust.
How robust?
Dixon and his team didn't just find this in one small group of people. They observed this split across 4 independent data sets, across 9 entirely different conditions, including just resting in the scanner, and 8 different active cognitive tasks.
Wow, so it's everywhere.
They found it at the level of individual participant data and even in meta-analytic co-activation patterns across thousands of prior studies. It's a very real divide.
It's undeniable. The management is truly split. Okay, so we have the workers on the floor, the DAN handling spatial focus, and the VAN acting as the circuit breaker.
Yep.
And we have our specialized middle managers, the FPCNB looking outward and the FPCNA looking inward. We know who the players are. But the million-dollar question that is bothering me is, how do they actually talk to each other?
Ah, the communication problem.
Yeah. What language are they speaking to coordinate all of this across massive physical distances in the brain in real time? Because earlier, you mentioned that a lot of this data comes from functional MRI or fMRI, and fMRI measures blood flow, right?
That's right.
But blood flow is sluggish. I mean, it takes a second for blood to move.
This is a critical point that often gets completely glossed over in pop science. fMRI is arguably the most incredible tool in the history of neuroscience, but it has a fundamental limitation. It captures the blood oxygen level dependent signal, or the BOL signal.
Walk us through what that actually means biologically.
Sure. When a region of neurons fires, it consumes oxygen. To replenish that oxygen, nearby blood vessels dilate, and a rush of oxygenated blood flows into the area.
Okay, makes sense.
fMRI magnets are tuned to detect the magnetic difference between oxygenated and deoxygenated blood. But that vascular response, the actual blood rushing in, takes about 5 to 6 seconds to peak after the neurons have already fired.
5 seconds. In brain time, 5 seconds is an absolute eternity.
Exactly. These bold D signals represent very slow oscillations, typically fluctuating between 0.0005 and zero point 0.1 hertz.
That's slow.
Very slow. That means you're looking at roughly 1 wave cycle per minute. But the cognitive constructs we're talking about today, shifting attention from a book to a crashing plate, realizing you need to change your internal goal, the FPCN sending a command to the visual cortex, these things happen in tens of milliseconds.
So fMRI just totally misses the action.
Pretty much. You cannot capture the speed of thought by watching the blood flow.
So if fMRI is too slow to catch the actual real-time conversations between these managers and workers, how do we eavesdrop on them? What tool do we use?
To understand the true electrophysiology of control, we have to look past the blood and look at the electricity. We'll look to a vital 2018 paper by Scott Merrick and Nico Dosenbach, published in Dialogues in Clinical Neuroscience.
Merrick and Dosenbach.
Yes. It's titled The Frontoparietal Network, Function, Electrophysiology, and Importance of Individual Precision Mapping. They synthesized decades of electroencephalography EEG and magnetoencephalography MEG data.
And those measure electrical signals directly, right?
Exactly. They wanted to figure out the actual radio frequencies these networks use to talk.
And what did they find? What is the frequency of cognitive control?
They found that the rapid coordination of the frontoparietal network occurs in a very specific, slower frequency band. Actual cognitive control and interareal synchronization of the FPN occurs in the theta and alpha rhythms.
Theta and alpha. So what numbers are we talking about?
We are talking about electrical oscillations in the 4 hertz to 13 hertz range, meaning the electrical wave peaks 4 to 13 times per second.
No, wait, 4 to 13 times per second is obviously faster than blood flow, but in the grand scheme of the brain, that's still relatively slow, isn't it?
It is relative.
Because I know that when neurons are doing intense local processing, they fire in the gamma band, which is incredibly fast.
You are absolutely right. Local sensory processing, like the neurons in your visual cortex, actively deciphering the curves of the letters on the page runs on gamma waves, and gamma operates at roughly 40 to 100 hertz.
100 times a second.
Exactly. So we have a paradox. The local workers are chattering away at 100 times a second, but the manager coordinating them is only speaking at 8 times a second. How do they communicate?
Okay, let me see if I can translate this electrophysiology into a metaphor to make sense of that paradox.
I'm for it.
Let's imagine the brain is a massive, sprawling city. The local sensory processing, that fast gamma wave activity, is like construction workers on a specific building site using high-frequency walkie-talkies.
Okay, I like this.
They are talking fast, giving rapid-fire micro-instructions. Move that beam left, drop that wire right.
Yeah.
It's great for rapid local communication, but a walkie-talkie signal is weak. It cannot travel across the whole city.
Right, it drops out.
Exactly. The frontoparietal control network, however, is the mayor's office. It needs to coordinate the whole city simultaneously. It cannot use a walkie-talkie, so it operates on theta and alpha waves, which are slower, but they act like a massive AM radio broadcast from a giant tower that can wash over the entire metropolitan area.
That is a phenomenal analogy. It perfectly aligns with the physics of neural coherence.
So the manager broadcasts this slow, steady, powerful pulse, the theta or alpha wave, and the local workers out in the visual or motor cortices somehow align their fast, frantic gamma walkie-talkie chatter to that slower broadcast.
Yes, exactly. In neuroscience, we call this phase-amplitude coupling.
Phase-amplitude coupling.
Yes, the amplitude, or the power, of the fast gamma waves is physically locked to the phase, the peaks and troughs of the slower theta or alpha waves. And your analogy hits on the exact biological reason why this happens.
Oh, really?
Yeah. The physics and biology of neural coherence over long distances literally necessitates slower frequencies. Slower oscillations have longer wavelengths. In the brain, signals have to travel down long white matter tracks, which are bundles of insulated nerve fibers, to get from the front of the brain to the back.
Right, the actual physical wires of the brain.
Exactly. And there are microscopic delays in that travel time. If the manager used a fast, high-frequency gamma wave to send a signal across the brain, the peaks and troughs would be so close together that by the time the signal arrived, It would be entirely out of sync with the receiving region.
It would just be static.
It would be complete static.
So the long, slow rolling waves of the alpha and theta bands are more resilient to the travel delays across the brain's physical geography.
Precisely. By using the phase of the slower theta and alpha oscillations, the frontoparietal network acts as a flexible, citywide hub. It essentially acts as a metronome.
A metronome.
Right. It tells all the local gamma band and walkie-talkies across the whole brain exactly when it is optimal to fire their signals so that the visual cortex, the motor cortex, and the attention networks are all communicating efficiently without talking over one another.
It is genuinely staggering to think about that level of electrical synchronization happening inside our heads just to execute the transition from reading a book to looking at a dropped plate in the kitchen.
It's beautiful, honestly.
It is. Millions of neurons aligning their firing rates to the trough of a slow AM radio wave broadcast by the prefrontal cortex. Okay, so we have built an incredible theoretical framework here. We understand the anatomical workers, the specialized managers, and the electrical radio frequencies they use to talk.
We've covered a lot of ground.
We have, but now I want to see this in action. Let's look at a concrete modern experiment that actually watches these networks physically reconfigure themselves when the brain is challenged with a complex task.
Okay, for that, we turn to a fascinating and highly complex 2023 paper in cerebral cortex by Moore and their colleagues. It's titled Functional Reconfiguration of Task-Active Frontoparietal Control Network Facilitates Abstract Reasoning.
Functional reconfiguration, okay.
Yeah. They wanted to see exactly how these control networks shift their allegiances when the brain is forced to stop resting and start thinking abstractly.
And to force the brain to think abstractly, they used a modified version of a classic cognitive test called the Raven's Progressive Matrices, right?
Yes, the RPM.
For you listening, if you have ever taken a formal IQ test or an aptitude test for a job, you have almost certainly seen a Raven's matrix. It is usually a 3x3 grid of shapes or patterns, but the bottom right square is empty.
Right, the missing puzzle piece.
Exactly. You have to look at the other 8 shapes and logically deduce the underlying rules of how they are changing. Maybe as you move left to right, the shapes rotate 90 degrees. Maybe as you move top to bottom, they change from black to white. You have to hold all those abstract symbolic rules in your working memory, synthesize them, and deduce which multiple choice option correctly completes the pattern.
It's intense.
It is. It is a test of pure, fluid intelligence and abstract puzzle solving.
Exactly. It requires intense cognitive control, visual attention, and working memory. So what Maureen and her team did was put subjects inside an fMRI scanner and give them a simplified computer screen version of this task to test both symbolic and perceptual reasoning.
While scanning their brains the whole time.
Right. The subjects were deducing these visual patterns and pushing buttons to submit their answers, while the researchers continuously tracked the functional connectivity across their entire brain.
So they're basically watching the high school cafeteria while the students are forced to take a pop quiz.
That is exactly what they're doing.
What did they discover?
They discovered a phenomenon they termed functional reconfiguration. When they analyzed the brain-wide connectivity matrices, they found that certain networks in the brain act as a stable, unchanging anchor.
Unchanging.
Yeah. For example, the somatomotor networks, which handle physical movement and touch, and the default networks, which handle internal resting thought, maintained a highly stable core community structure. The groups of voxels that talk to each other during rest continued to talk to exactly the same voxels during the difficult puzzle. Nothing really changed in their organization.
They were rigid, but I'm guessing the managers were not.
Precisely. The frontoparietal networks, including our FPCN managers and our Dorsal Attention Network workers, did not stay stable at all. They flexibly reconfigured their functional connectivity.
How so?
They dropped connections with certain regions and rapidly forged new temporary connections with other regions specifically to solve the matrix puzzle.
So while the rest of the brain was relatively static, the frontoparietal networks were basically rearranging the furniture in real time, bringing different experts to the table depending on what the specific visual puzzle demanded.
Exactly.
How on earth did the researchers measure that kind of fluid, dynamic shift across thousands of data points? I mean, that sounds impossible.
It's definitely difficult. They utilized a brilliant mathematical approach called the Louvain Community Detection Algorithm.
Okay, we definitely need to break that down. How does the Louvain algorithm work?
Well, it's an algorithm designed to extract the community structure of large networks. It was originally designed to study complex systems like social networks on the internet or the spread of epidemics across a country.
Oh, interesting.
Basically, you give the algorithm a massive hairball of connections, in this case, thousands of brain regions, and how strongly their these signals correlate with one another. And the algorithm's goal is to partition those regions into distinct communities so that the connections inside a community are maximized and the connections between different communities are minimized.
Okay, going back to our cafeteria analogy, the Louvain algorithm is looking at thousands of interactions and drawing circles around the tightest friend groups. It ensures that the people inside the circle talk to each other way more than they talk to anyone outside the circle.
Exactly. That's exactly. By applying this algorithm continuously as the subjects transition from just resting in the scanner to actively performing abstract reasoning, Morin and her colleagues could mathematically prove that the boundaries of the frontoparietal communities were shifting.
The clicks were changing.
Right. The FPCN was pulling in nodes from the attention networks, integrating visual data, and then releasing those nodes when the trial was over. This flexible reconfiguration is what puts the brain into what the researchers termed a task-ready state.
Task-ready?
Yeah. It primes the specific necessary regions to activate efficiently for unique, complex cognitive demands. The FPCN isn't a static manager. It's a fluid task force.
But wait. If the frontoparietal network is constantly reconfiguring itself based on the task, if it's constantly pulling in new nodes and dropping old ones, how can neuroscientists ever draw a definitive map of the human brain?
That is the problem.
It feels like trying to map a city where the streets literally change direction and intersections move depending on what time of day it is. How do you create an atlas for something that fluid?
You have just articulated the central crisis, and really the most exciting frontier in modern cognitive neuroscience.
Oh, really?
Yes. The realization of this extreme fluidity and complexity is leading to groundbreaking and, frankly, humbling recent discoveries. Our understanding of these networks is evolving so rapidly that the very names we use for them are being rewritten, and the fundamental way we scan and map brains is undergoing a massive paradigm shift.
Okay, let's start with the names being rewritten, because in science, you don't just change the name of a brain network unless you fundamentally misunderstood what it was doing.
That is exactly right. In early 2024, Nico Dosenbach and Marcus Rachel published a major perspective piece in Nature Reviews Neuroscience titled The Brain's Action Mode Network.
The Action Mode Network.
Yeah. Yes. For years, a key cognitive control network, one that works alongside the FPCN to maintain goals, was anatomically labeled a cingulo-opercular network, or CONN.
CONN.
Right. It was named purely based on the physical folds of the brain where it resides, the cingulate cortex and the frontal operculum. But Dosenbach and Rachel argue this network needs to be functionally re-annotated as the Action Mode Network, or AMN.
Oh. Why the rebrand? What does action mode signify that cingulo-opercular misses?
It represents a fundamental shift in how we view the macro states of the entire human organism. The researchers argue that the Action Mode Network sits diametrically opposite the default mode network.
The daydreaming network.
Right. They counterbalance each other like a grand, brain-wide yin and yang. While the default mode is active during self-referential internal resting states, when you are safe and relaxed, the AMN prepares the entire organism for action. It is the network that shifts your whole being from rest and reflect to ready to act.
So it's not just a cognitive process, it's a full-body physiological state.
Entirely. When you enter the action mode, it isn't just about thinking harder. The AMN heightens your physiological arousal. It focuses your attention externally. It creates action plans and continuously updates them based on feedback. It is responsible for processing instructional cues and sustaining goal maintenance. But incredibly, it also integrates action-relevant bottom-up signals, like physical pain or visceral sensations. It even provides sympathetic drive to your internal organs.
Sympathetic drive, you mean the sympathetic nervous system, the fight-or-flight response.
Yes, exactly. The AMN is physically wired to trigger that drop in your stomach feeling, the increase in heart rate, the slight sweating of your palms when you realize you just made a critical mistake on a project, or when you're preparing to execute a difficult physical task.
So it's fully connected to the body.
Exactly. By renaming it the Action Mode Network, the field is acknowledging that cognitive control isn't just a disembodied computer program running in the cortex. It's intimately tied to the survival and action readiness of the physical body.
So we are moving away from naming things based on their anatomical zip code and naming them based on their global whole organism behavioral purpose. But you also mentioned a paradigm shift in how we map these networks. You were talking about the end of the group average in fMRI studies, aren't you?
I am. And this is perhaps the most consequential shift for the future of clinical medicine.
Really? How so?
Well, for decades, the standard fMRI paradigm was to scan a person for maybe 5, 10, or 15 minutes while they did a task, because the fMRI signal is incredibly noisy. It's corrupted by the subject's breathing, their heartbeat, minor head movements. Researchers would take the scans of hundreds or even thousands of people, align their brains to a standard template, and average them all together to find the typical brain map.
Right, the idea being that if you average 1,000 blurry pictures together, the noise cancels out, and you get one clear picture of the true, universal human brain.
Exactly. And it worked incredibly well for mapping basic sensory and motor functions. Everyone's visual cortex is in roughly the same place at the back of the head. But a landmark 2017 paper in Neuron by Evan Gordon and colleagues, titled Precision Functional Mapping of Individual Human Brains, proved that this averaging method actively obscures critical life-saving information when it comes to the complex association cortex.
The front of the brain.
Right, where cognitive control happens. They introduced a project called the Midnight Scan Club.
The Midnight Scan Club. I absolutely love that name. It sounds like a secret society of sleep-deprived researchers doing rogue science. What did this club actually do that was so revolutionary?
Well, the standard practice was 10 minutes of data per person. The Midnight Scan Club, led by researchers at Washington University, scanned a small group of highly dedicated individuals for 300 minutes each.
300 minutes. That is 5 hours inside an fMRI tube.
5 hours of resting state data per person, collected across 10 different sessions, often late at night when the expensive scanners were free and unused, hence the name.
That sounds brutal.
If you've ever been in an MRI, you know it is not easy. It's a tiny claustrophobic tube, it's incredibly loud, and you cannot move your head even a millimeter or you ruin the data. But by collecting such an immense, unprecedented amount of data on single, specific individuals, they were able to mathematically overcome the high noise-to-signal ratio of fMRI without needing to average it with anyone else's brain.
Oh, wow.
They could create a pristine, high-resolution map of one specific person's brain.
And what happens when you look at a pristine map of one person instead of a blurry average of 1,000 people?
The revelations were just staggering. They proved that while the core network components are indeed shared across humanity, individual human brains have radically different functional topographies, particularly in the prefrontal cortex.
Topographies meaning the actual physical location of the networks on the folds of the brain.
Yes. What looks like the frontoparietal network in one person's brain might be mapped to a physically different anatomical fold in another person's brain. It's like looking at a map of the United States, and in your brain, Chicago is in Illinois. But in my brain, Chicago is functionally located in Ohio.
That is insane.
And it goes further. To quote the Merrick and Dosenbach 2018 paper summarizing this shift, they found that variants of other control and attention networks are located in regions of frontal cortex affiliated with the frontoparietal network in other subjects.
Wait, let me make sure I'm understanding the gravity of this. You are saying that if I point to a specific square inch on my forehead underneath that bone, that patch of brain might be running my outward-facing FPCNB.
Yep.
But if I point to the exact same anatomical coordinate on your head, your brain might be using that patch to run your default mode network or your ventral attention network.
Exactly. The functional real estate is allocated completely differently from person to person.
Yeah.
The group average maps we relied on for decades essentially blurred all these unique individual variations into one generic blob that doesn't actually represent any real human being's actual brain.
But if that is true, the clinical implications are completely massive. Think about psychiatry and neurology. If you are trying to treat a severe brain disorder and you aim a physical treatment at the average brain coordinate you found in a textbook, you might miss the patient's actual target network entirely.
Precisely. This extreme individual heterogeneity has profound implications for psychopathology and targeted therapies.
Like what kind of diseases?
Diseases that heavily involve cognitive control and executive function deficits, like schizophrenia, severe anxiety, major depressive disorder, and ADHD, all show profound disconnectivity in these precise frontoparietal networks. For instance, in schizophrenia, patients demonstrate marked, measurable reductions in activity and connectivity within both the frontoparietal control networks and the newly termed action mode networks. It is a core feature of the pathology.
So how do we treat it if we don't know exactly where the network is?
Well, historically, we use treatments like transcranial magnetic stimulation, or TMS.
TMS.
Right. TMS uses powerful magnetic fields placed against the scalp to induce electrical currents inside the brain, stimulating or inhibiting specific nerve cells. For a long time, doctors relied on group average anatomical coordinates to place the magnetic coil. They would measure a certain distance from the bridge of the nose, place the coil, and fire.
But based on what you just said, that's a huge gamble.
It is. If the target network, say the FPCN, is displaced by even a centimeter in that specific patient, the TMS pulse is hitting the wrong network. It might be hitting the default network instead.
Which explains why some people respond miraculously to TMS and others see absolutely no benefit. We were essentially firing blind based on a generic map.
Exactly.
Yeah.
The future of clinical neuroscience, born from the Midnight Scan Club and the realization of individualized topography, is precision mapping. If we want targeted neuromodulation therapies to work, we must put the individual patient in the scanner, collect enough data to overcome the noise, and map their specific, unique frontoparietal control network before we ever try to stimulate it. We have to map the territory before we intervene.
Wow. This has been an absolutely phenomenal deep dive. We have covered a vast amount of complex, cutting-edge science today. Let's do a quick structural recap to tie all these intricate threads together for the listener.
Sounds good.
We started with the fundamental hardware of how we process the world. The Dorsal Attention Network, or the DAN, acting as our top-down, spatially mapped spotlight, physically moving our eyes and tuning our visual cortex to keep us focused on our internal goals.
Right.
Then we looked at the right lateralized ventral attention network, the VAN, which uses complex dynamic causal modeling to evaluate the environment and act as a crucial circuit breaker, alerting us to sudden, salient changes.
And from there, we scaled up the hierarchy to the middle managers,
Yeah.
the frontoparietal control network. We saw how machine learning algorithms revealed that this network is brilliantly divided into the outward-facing DAN-connected FPCNB and the inward-facing default network-connected FPCNA.
Then we solved the speed problem. Because fMRI bold signals are too sluggish to track real-time thought, we looked at the electrophysiology. We explored how the managers communicate across the massive physical distances of the brain using long, slow theta and alpha waves.
The AM radio broadcast.
Exactly, acting as a citywide AM radio broadcast that synchronizes the high-speed, local gamma wave chatter of the sensory workers.
We also looked at the fluidity of the brain. We saw through the Morin 2023 paper and the Louvain community detection algorithm how these frontoparietal networks dynamically drop and form new connections to physically reconfigure themselves into a task-ready state for abstract reasoning.
And finally, we uncovered the highly individualized, unique topography of our own brains. We saw how networks like the newly rebranded Action Mode Network, which physically preps our entire body for action by triggering our sympathetic nervous system, are functionally powerful, but anatomically unique to every single human being, completely upending how we will treat psychiatric disorders in the future.
It really changes everything.
It does. So for you listening, I want you to remember this massive biological symphony the next time you're sitting at your desk trying to focus on a dense spreadsheet. When you get distracted by a pinging phone notification and you have to force yourself to pull your eyes away from the screen, ignore the phone, and get back to work.
It happens to all of us.
All the time. That incredibly mundane, everyday struggle is actually a lightning-fast miracle. It is your VAN breaking the circuit, your FPCNB evaluating the distraction, your theta waves pulsing across your cortex to realign your attention, and your DAN physically wrenching your eyes back to the spreadsheet. And it is all happening on a topographical map inside your skull that is as unique to you as your own fingerprint.
And as we close, I will leave you with a final thought to mull over, looking toward the horizon of this science. We now know that our cognitive control and attention networks are functionally reconfiguring themselves all the time to put us in a task-ready state.
Right.
We also know that we are developing the technology to precision map these exact networks down to the millimeter in an individual person. So the question becomes, could we eventually use advanced real-time biofeedback or perfectly targeted, non-invasive neuromodulation to consciously induce this action mode at will? What are the profound implications for human potential, for education, and for creativity if we develop the safe technology to manually flip the switch on our own dorsal attention network whenever we choose?
A completely functionless ability to enter a flow state on command. That is a tantalizing frontier to ponder, and it entirely changes how we think about the limits of human focus. Thank you for joining us on this exploration of your own mind. Keep your curiosity sharp, and we will catch you on the next deep dive.